All right, thank you very much. Thank you for this kind introduction. It's a real honor to be here and give this talk. I was just telling Andreas, this is actually I'm ashamed to say my very first time attending this conference, and it's really interesting. I hope to come back more frequently. So there is really no better time to talk about systems modeling than today. The pandemic has shown us that there are tremendous opportunities for modeling. It has also shown us that there are tremendous pitfalls for modeling if we're not careful, and there's lots of challenges with this convergence of mechanistic modeling, statistical modeling, and especially artificial intelligence. I think since this session is focused largely on mechanistic modeling, that's what I will talk about. But I think bringing together artificial intelligence, data-driven modeling, and mechanistic modeling is one of the great challenges and opportunities that we collectively need to address. So what I will tell you today is about some of the models we've worked on for some time now, and what we plan to do with them, and then take this model as an example for some of the broader questions that we face, things like model selection, or things like, how complicated or how simple should models be. And I have thoughts about these, and I have beliefs about them, but I think there's a lot of different points of view one can have, and I hope to really make this largely a basis for a discussion which we can have either here following or subsequently. So just to get the acknowledgements out of the way, this is a project that's been going on for some time, and it has involved quite a number of people, in particular my lab, both at the University of Connecticut and at the University of Florida. The Merad lab, so Borna Merad is my longtime collaborator. He's a pulmonary epidemiologist. He's the director of the pulmonary ICU and division chief at the University of Florida, and he sort of grounds this whole effort in the right scientific and clinical framework. And we've been working with a company, Kitware Incorporated, a tremendous resource in data science and scientific visualization. And of course, we were lucky to have significant support from different funding agencies. This picture is part of my lab. It's a hike last July in the middle of Florida in an alligator preserve, where you can get within 20 feet of an alligator, which is about as close as you should get. Maybe not even that close, but it was an amazing place, and it's amazingly hot and humid. So what I want to tell you about is a modeling project that we had focused on fungal infection, respiratory fungal infection. So here's an article some years ago in the Atlantic 4 years ago, and it has a story about. It starts with a story about a patient in a Dutch hospital that was immunocompromised and acquired an infection with a fungus Aspergillus fumigatus, got invasive aspergillosis, and this fungus was resistant to frontline antifungals obtained. So one of the causes for strains that are resistant to antifungals is same problem as with antibiotics and the animal industry. With antifungals, it is antifungals and the agriculture industry. So in this case, it turned out the tulips in the hospital garden were full of Aspergillus spores with antifungal resistant strains. So that's a big problem. The problem is exacerbated because immunocompromised patient populations are growing. Every time you undergo chemotherapy, you're severely immunocompromised and in danger of an infection with this fungus that can happen with as few as a handful of spores that you inhale. It has a very poor prognosis, has a mortality of almost 50%, even with frontline treatment. So our goal was to use computational modeling to focus what the community is focusing on over the last several years, and that is to look at ways to help the immunocompromised host and its immune system to fight the fungus rather than just focusing on fungus-centric treatment. So that was the motivation for us to get into all of this. So just to recapitulate the biology of it a little bit, it's fundamentally a multiscale problem. What you see in the left panel is essentially we inhale airborne conidia. They're everywhere. This fungus grows on decaying plant materials and so forth. So we all inhale hundreds of these spores every day. They come all the way down to the lower respiratory tract where if, as I said, if you're immunocompromised, then there's a possibility that they will lodge in your lower airways, germinate, and cause all sorts of problems. What the body does, part of the immune system, is primed to react to that. As soon as epithelial cells, as soon as resident macrophages detect the fungus, a cascade is set in motion that gets these cells to secrete IL-6. IL-6 gets transported to the liver. The liver synthesizes a hormone hepcidin. Hepcidin gets transported back into the lungs and reduces the propensity of cells to export iron, thereby depriving the fungus of iron, and iron is a key nutrient for fungal virulence. So that's the particular feature we focus on. Of course, there's any number of other things that are happening. What you see in the middle panel is what happens sort of at the tissue level. You have from the circulation through various signaling events, you have different immune cells entering, in particular neutrophils, and neutrophils are key immune cells to help you fight this fungus. And for instance, if you're undergoing chemotherapy for cancer, then over a period of days, your supply of neutrophils is essentially wiped out. So that is the key. What you see in the right-hand side is the mechanism for iron export, in this case in macrophages. One of the jobs of macrophages is to scavenge dead and dying red blood cells, so they're full of iron. And here you see the mechanism that hepcidin uses to essentially interrupt iron export by deactivating the receptor on the cell surface of macrophages. So that's the basic biology, the basic immunology that goes into this, and that's essentially what we model. And after a number of years working on this, we just recently, a few months ago, published this paper that contains the model. And we used it, our first use case for this model was to look at what happens with fungal strains that have different virulence. Which parts of the immune system are engaged from the point of view of virulence? And what we show in the paper is that for different levels of virulence, different parts of the immune system are most responsive. So you see in the title the two key features are leukocyte activation and iron acquisition. So I want to tell you something about this model and then tell you what are good things about this model and maybe what are not so good things about this model. And we can lead to a bigger discussion from there. So here's a little diagram that indicates the different mechanisms that are built into this. And I should say before I describe it, so this has an intracellular level. Macrophages, fungal cells, they carry with them intracellular networks that essentially a macrophage, let's say, finds itself in a different part of this physical space, and it decides what to do using this intracellular decision network, which is in the form of a Boolean network similar to fungal cells. So fungal cells in particular, we focus on iron acquisition. If a fungus requires iron, they will synthesize certain molecules called siderophores that go out and essentially scavenge for iron. So those are Boolean networks. At the tissue scale, it is an agent-based model, and one reason why we chose an agent-based model is that it's, as I said, this infection can get started with a small handful of spores, so it is very, very heterogeneous spatially. So you have these clusters of fungal growth, which would be quite difficult to capture with a continuous model, say a PDE or a compartmental ODE or something along those lines. And then there are simple transport equations that bring hepcidin from the liver and bring IL-6 to the liver and so on. So that's the computational structure of the model. What it looks like is, so here you have fungal cells come in, they start to germinate and grow into hyphal structures. They are recognized by a number of different cells. They're recognized by resident macrophages. They're recognized by epithelial cells. And then these cells secrete cytokines and chemokines, most importantly IL-6. And IL-6 then activates processes in the liver, which then reduce the ability of the fungus to scavenge iron from the host. It's a fairly complex model. It has on the order of 75 parameters. It has many state variables. It has, I think, 15 or 16 cytokines and chemokines. Cytokines and chemokines and iron are modeled with partial differential equations as they diffuse through the system. So it is a hybrid model. And we also have a spatial version of it. So it runs in a spatial version. I'll show you a representation of that in just a minute. And I'll tell you a few more things about this model as we go on. Here are some of the intracellular models. As you see, the model for the fungal siderophore synthesis and secretion we published some time ago. That was validated by experimental data from a colleague of mine at the time at Virginia Tech. When I was this model for macrophages should appear any day now, I hope, and that was built. So it essentially captures what a macrophage does when it comes into contact with a fungal structure, and it was built partially with the literature and partially from an RNA-seq data set that we collected with primary human macrophages that we exposed to Aspergillus, and we collected data over time, and then added some RT-PCR validation experiments. So that was the basis for this model here, and the macrophages in the model I showed you previously carry some modified version of this along with them. We're working now on intracellular models for neutrophils, for dendritic cells, for some of the other cells in particular, and I'll get to that in a little bit, endothelial cells, because the real danger with this particular fungus is that the fungal structures grow through the epithelial layer of the alveoli, through the interstitial space, through the endothelial layer of the blood vessels that surround the alveoli, and then it becomes a systemic infection, at which point it is quite dangerous. So these are the intracellular models. This is the spatial representation. So this is a finite element method rendition of what you see here. These structures here represent alveoli. They're clustered together along an alveolar duct. That's this purple structure here. You can see over here a representation of diffusion different molecules that's implemented. Now, from the point of view of looking realistic, this is not very good. From the point of view of, for example, doing fluid dynamics, it will also be terrible. From the point of view of the dynamics that we needed to support, it is quite sufficient. One could make it much more fine-grained, which would add computational complexity. But in this case, not necessarily faithfulness to what's going on. And we have a number of other features, like essentially dashboards for experiments. So this is really the advantage of working with a consumer of models like this, is that you get a dose of reality when you ask people to interact with these models. So we try ultimately to build these models so they can be interacted with by an immunologist, essentially. Now, how do you validate a model like this? It is reasonably complex. So the first thing we did is we parameterized this model, not by data fitting. We parameterized it by finding values for all 75 parameters in the literature. Now, in this case, that's possible because the literature in this field is very, very extensive. People have measured lots of things, and so for some of them, we had to, maybe take some experiments and extrapolate some things, but basically we found parameters in the literature values for all of these. And so then what we did is we took these 8 data sets. So these are data sets in the literature that we didn't use to find parameters. And what we showed, and you can see here in this table, they're quite varied. They're all mouse experiments. So our model is really calibrated to a mouse. And the experimental system we have is a mouse system. Essentially, it's a system in which you can induce a transient neutropenia. So you can eliminate neutrophils for a period of days, which would simulate, let's say, somebody undergoing chemotherapy. And so we took these 8 experiments and we showed that with these default parameters that we picked, the model qualitatively fits each one of these data sets. And here are some examples. So you see, here's, for example, number of neutrophils, I think, so neutrophil population over time. Now, obviously, you won't get good numerical fits. What is important in this case is that you get the shapes of the curve correct. What is even more important is that one gets the timing correct, because as I said, the model is an agent-based model, so there's no real time built in. So the time that this model has is essentially implicit in what happens between one time step and the next. So the fact that the timing is quite accurate is, we were very, very happy about that. And so again, this is a fit with the default parameter set. So there's no fitting of any sort that's going on. The other thing we did is we did a lot of parameter sampling around this default parameter set to show that, in fact, you can reproduce the variation that you see in these different experiments with the model. So the model accurately captures variability. And then the other thing we did is we performed ourselves an experiment by taking this mouse model and exposing it to neutropenia over a course of, as you see here, 72 hours. And you also get good qualitative agreement, with the exception of actually neutrophils. So here, our neutrophil count is quite off. The interesting thing is the measured neutrophils are actually quite off from other experiments. And so the experiments that I showed you in the other papers. And so there's some question over how to interpret this particular data point here. But in a nutshell, again, with the default parameters, we found this model very well qualitatively captures a range of different experiments. this particular condition. Now, what other things have we done with this model? Right, so we've built the model, so what do we do with it? And I'll get back to that in a bit, but one of the things we've done is looked at a phenomenon that appeared recently. So invasive aspergillosis is a phenomenon for immunocompromised patients. Almost nobody who is immunocompetent will get sick with invasive aspergillosis. However, what happened is over the last couple of years, there has been a number of COVID patients who were immunocompetent and still got invasive aspergillosis. And so what we did is we used this model to essentially look at what are the potential mechanisms that are responsible for that happening. Can we identify particular mechanisms that are responsible and particular parameters that are responsible for this dual infection? And in fact, one can within a few days, this paper will go up on the bioRxiv, so you can read the details of it. It's quite interesting, and the distribution. So the way we do this is we create a virtual population of about 25,000 patients through parameter sampling, and then look at various outcomes, and we create a distribution of patients that are infected with one or both of these pathogens, and then study the mechanisms that go with them. So that's another application. And then finally, a third application that we're working on quite actively is based on a discovery we made a little bit ago that we published just a few months ago, earlier this year. And that is that Aspergillus, the fungus, can actually utilize extracellular heme. So there's an additional ion source that in particular happens when hemorrhaging happens in the aftermath of the infection. And what was shown in the lab is that in fact Aspergillus can take advantage of that source of iron, and it does affect the virulence and infection and severity of the infection. So that led us to looking at a particular observation, the simple observation that if I delete neutrophils from the patient, then macrophages alone almost never are able to control this infection themselves. So the question is why? And the hypothesis that we have, of course, is that it has to do with, in particular, two effects of hemorrhaging. One is that hemorrhaging changes the phenotype of macrophages. It makes them less angry, it makes them less aggressive, and it makes them less effective in doing what they're supposed to do. And it provides an additional iron source for the fungus. So these two together, in some combination, we hypothesize support this phenomenon that neutrophils are needed. And so if that turns out to be correct, which certainly from the point of view of the simulations, it seems plausible, and from the point of view of the experiments, to the extent we've done them, that seems plausible. Then one could think about, what can one do in order to affect hemorrhaging, either delay time-wise, reduce its effects, and so on, to sort of help macrophages to do their job. And that was, that's the sort of thing we were hoping to do at the very beginning, as I mentioned, when we started this process. Okay, and so now, having done this, we're working on expanding this model. Fungal pneumonia, which is what this essentially is that we're talking about, is relatively rare in comparison to bacterial pneumonia or in comparison to viral pneumonia. And so we want to expand this model to cover all types of pneumonia, fungal, bacterial, and viral. So what you see here is schematic of endothelial cells we're going to add, of neutrophils we're going to expand. We're going to expand the intracellular network for epithelial cells, the same for macrophages, to capture some of the features of a virus, in this case SARS-CoV-2, interacting with these different cell types. So that's how you should read this picture. It's not a sort of a compartmental model of some sort. It simply is a way to show which agents are included and how these agent types interact with each other. So that's where we're going now. Now, endothelial cells are really, really important. The fungus modifies gap junctions between endothelial cells. That's what causes leakiness, and that's a phenomenon that actually is quite common. It's an effect that one sees in diseases like pulmonary hypertension and diseases like that one sees in relation to lung transplantation. So epithelial cell biology is really, really crucial for this. Okay, so complex as it is, we're going to make it much more complex. And let me just point you to a method we have to do this. So we've worked quite a bit on this idea of how do you expand models? How do you modify models without continually having to tear down what you have and starting from scratch? And anybody who has built complex models knows you want to try to be modular, but what you end up having is a bunch of modules that pass data between themselves, and if you change modules, if you add modules, if you delete modules, and you try to then rerun it, more often than not, it'll break, and it's very difficult to test. So how to make this modular? Well, what I think one cannot do is make mathematical models modular. They are what they are, and the different variables simply depend on each other. So what we've done is, instead, we have tried to make the simulation model modular by creating a hub and spoke architecture where the hub is the data, right? So what you see here, this thing in the middle here, is the data space. It has all the data in it that you have that describe the physical space, that describe all the different agents, all the different concentrations. It actually has in it all the parameters in all the modules. So all the data are in there. None of the computational logic is in there. All of the computational procedures are in these modules here, and nothing else is in these modules. So no data are in these modules. And these modules do not talk to each other. All they do is they modify certain parts of the data space. And the way two modules do interact, so a macrophage will engulf a fungal spore, so they have to interact, but the way they interact is by modifying overlapping parts of the data space. And so in this case, if I take one of these modules and I pull it out and I replace it by another one, the only thing I need to worry about is how this module interacts with part of the data space. As long as I preserve that, I can do in this module whatever I want. It will not break anything. That's really the basic idea behind this. Okay. Now, in the last 10 minutes, let me put this in context a little bit. So it's a complicated model. It's stochastic, it's hybrid, it's multiscale. So there's any number of computational and mathematical issues that go with it. In terms of validation, we've done a number of things. I told you about some of them. We have parameterized it with the literature. We've shown that it quantitatively fits data sets. We've shown that one of the things you'll see in the paper is that the difference between the simulated data and any of the data in this in these other data sets are comparable. We've done a number of different things to validate that. So from our point of view, it's a model that we and our collaborators trust. Now, the question that models like this face oftentimes is this the correct model? And we have faced that when we tried to submit papers, when we tried to submit grant proposals. Is this the correct model? So how does one answer that? Well, so if you use the quote, I don't actually like this aphorism, it's almost, that all models are wrong, but some are useful, by George Box, then well, then the question doesn't apply. It's not the correct model. No models are correct. Of course, what Box concluded after that is don't worry about models being correct. Only worry about models being useful. And if you want to do model selection, then choose a parsimonious model. That's the rule. But I think that more importantly for us, the models we build, they need to be credible. That is a lot of the usefulness. And credibility is determined by the users of our models, not by us. Okay, so that's, I think, an important criterion. Now, what do we want to do with this model and subsequent models? Is we want to do two things. Is we want to do studies like the three that I showed you, generate scientific hypotheses that you could test. But a little bit more, or much more ambitiously, is we would like to take models like this, and we would like to personalize them to patients, and we would like them to be used. And in fact, to me, that's an overarching challenge, and maybe one of the most important ones that we could face. And from that point of view, all I care about is, and all the clinicians care about that I will go to, is whether they think this model is credible. And that's when anybody would give you the time of day. Now, the other argument for models of this type is, I think, that complex systems need complex models. And we just need to go with that, right? There are some examples of complex model projects, virtual physical human, virtual liver, and so on. I think if we want to build models that have impact, we should look to science and areas that have had tremendous impact, like weather forecasting. Right? I live in Gainesville, Florida. If there's a hurricane brewing in the Gulf, I want to know, is that hurricane going to hit us? Should I pack up my car and leave? It's a very important question, and that is all mathematical models. What I want to propose, and we did this in a perspective we wrote some years ago, I want to propose a complementary modeling approach that violates sort of all the modeling guidelines that we have been following. And the first one is build the most complicated model you can. Throw in everything you know. Right? We thought about agent-based models. It's very easy for domain experts to use agent-based models. You can add everything in to the point where the model contains everything you know about the real world that you're interested in. And you know, our model that I showed you is sort of a pathetic attempt at something like that, you know. And so you can treat that as a surrogate for reality. So then when you have a given problem of interest, then see what in this model is important, what's important in the real world, which variables, which parameters. Then you pick those. And as I've shown you in those three examples, if you actually look at the papers, you see in each case, there's a small number of state variables and a small number of parameters that are relevant. And then you can go and you can model the model. You can build a model of that part of reality that you're interested in. And we've worked on this quite a bit. We have a manuscript coming up that focuses on approximation of agent-based models with a particular control problem by ODEs and then doing control. So, so that's when we submitted this paper first. One of the reviewers said this is simply a terrible excuse for poor modeling if you try to do this. But I argue that it's actually a very efficient way of going about it. So with that, I thank you.
Thank you so much for this wonderful talk, Dr. Laubenbacher. could you please stay on the stage for a few more questions? We have only one microphone, so I keep the mask on to not expose you to anything. So I'm sure there are questions from the audience. Do I see someone raising their hand? No, it's okay. No questions at this time. Oh, there is one. Okay. Oh, I think there's a microphone in the middle. Maybe you want to use that one?
It's really tall for me. But thank you for the fantastic talk. A lot of your models based on evidence coming from mouse models. So how is it translated into human models generally?
Yeah, so that's of course, there are several aspects to this. One is simply a matter of allometric scaling, right? The lung of a human is bigger than the lung of a mouse. There's more macrophages, more other things. So a lot of parameters will change. And certainly humans have some key aspects of their immune system that's different. Lung physiology is different, so that needs to be built in. But if you want to build a generic model, then some animal model is the place to start. Otherwise, you have really no ground truth. There are many experiments you can do with mouse that you can't do with humans. So that's where we are. The next step for us, actually, what we're doing is we have a tissue culture system that allows us to use primary lung tissue from surgeries that one can infect, and you can do microscopic microscopy studies. You can do single cell transcriptomics on them. So that for us is the next step to translate to human.
Talking about this is really something I also wanted to ask. So because you said you collected many parameters from the literature, how well did they fit? Isn't it sometimes happening that measured value in one experiment isn't compatible to others? Did you experience that, and how did you deal with that?
Yeah, so certainly you can find some parameters, you can find more than one value in different experiments, and then it's essentially a matter of judgment in which one you use or how you might average these things. So there have been some cases we've documented that quite carefully, and it's important. On the other hand, again, for us, this parameterization is essentially a reference parameterization, sort of like Craig Venter's genome is for the Human Genome Project. Right? So it by itself, I mean, we don't think of it as the correct model. It is simply a model that provides a reference.
Hi, I'm very interested in your the model idea, and I was wondering if you could just elaborate on that a little bit more. Tell us what it might look like in terms of, what the elaborate model might look like, and then how you would kind of abstract from that.
Right. So we're looking at a number of agent-based models. We've done this largely in the context of heterogeneous biofilms, but we're doing it in the context of this model and some others. The idea is you specify a control objective. We did this specifically in the context of control. Specify a control objective, and then you build an ODE model, and depending on the features of your ABM, there are a number of different methodologies you will need to use. So if you have a steady state, you can approximate around the steady state, let's say, if you want to move a steady state or various other features. And then you optimize the ODE that you collect from the point of view of the control problem. So the goal is not to build ODEs that dynamically capture the ABM. That is not of interest. The interest is the control problem, and once you can solve this in your ODE setting where you have tools, then you can lift that to the ABM.
Let's allow one quick question and then.
Just a quick question. Nice talk. So we are in an era where there's a lot of generated data on single cells and things like that, and there's an increase in complexity and heterogeneity. So can you comment how that complexity can be fitting into the proposed model?
Yeah, so as I mentioned, as part of the answer to the previous question is, with the human tissue cultures we have, we will collect single cell data, because that's actually the only thing you can do. There's no, it doesn't make sense to collect anything else. And then those single cell data will be fed into the construction of the model. For some things, single cell data are important. For other things, I'm not so sure, but in this case, they certainly are.